Hi everyone, this is Jenna. I hope you are all doing well and adjusting to sort of this new normal that we're all experiencing. As I've told you in emails, I'm going to post a new lecture once or twice each week to help you get through the material that would have been part of our molecular biology of the gene course and actually is still going to be part of our material. Originally, this lecture for this week, we would have been dealing with things such as mutations and genetic variations, but I think this is a really good time for us to move forward our lecture and sort of exploration of viruses. So that is what we're going to do here today. And then this will be followed by a lecture in the next couple of days related specifically to coronavirus. I know we've talked a lot about it in class as we saw the impending pandemic. However, we're going to get more into what is our current knowledge of this virus and how do we study it, etc. Okay, so let's get going. So what are viruses? Well, all of you guys know that they're essentially infectious agents. Viruses underlie many of the common diseases that we are all either exposed to at one time in our lives, or perhaps we've read about in the news, or have seen someone else struggle with. One of the biggest questions are they actually alive? As we've discussed in class, they really are not alive, even though we may discuss them as if they're a living thing, talking about how long does it survive on a surface or where it moves around, etc. Remember from cell biology that we have 7 criteria that must be met in order for something to be considered living. The animal or living thing in question, it needs to take in nutrients from the environment and use those nutrients to grow or produce energy or something else. They need to show sensitivity to stimuli and respond to that stimuli, very much the way that we might move from a cold area to a warm area, or a plant might adjust itself to maximize its leaves' abilities to take in the sun. Things that are alive must reproduce, and please notice that I have that in bold. We will discuss that in a minute. And that essentially is making more of themselves, whether that is a single cell dividing to make 2 daughter cells, or whether that is an entire organism making more of that same organism. Living things grow. They take in nutrients and they make more cells, or they increase in size. Living things move. They also excrete things. They get rid of their waste products in a way to keep their bodies healthy, and they undergo respiration. So again, that is that release of energy from food or nutrition sources. Now, in reality, viruses don't meet at least 6 of these 7 criteria, right? The only controversial one is, do they reproduce? Some people would argue that yes, they do meet the condition that they reproduce, and so therefore they satisfy that criteria. Others will say, well, they don't really reproduce because they have to hijack another cell to make more of themselves. I tend to fall out on the side where they don't really reproduce in the typical way, but some people believe they do. And that is the only criteria of living things that viruses do actually meet. Now, when we talk about that reproduction, again, they don't reproduce in the way of being able to do this themselves. They have to hijack another cell in order to make more viruses. And the types of cell types that they hijack are very specific. Viruses don't just bind to any specific cell type and infect that cell. They have what we refer to as tissue tropism when we're talking about eukaryotes, meaning that they target specific cell types. Now, you'll remember from our discussions in class that if viruses target bacteria or prokaryotes, they are called bacteriophages. Those are very, very specific viruses that target bacteria. If viruses target eukaryotes, we then refer to them strictly as a virus. In either case, whether it is a bacteriophage or whether it is a eukaryotic cell, the specific virus will seek out very specific cell types, which it will recognize on the basis of specific receptors or molecules on the surface of that cell. So let's talk a little bit about bacteriophages. In addition to being sort of a really, really interesting clinical strategy for treating certain bacterial infections and possibly being sort of our next step if we eventually have a full failure of antibiotics, they have very specific characteristics that we recognize. So you'll remember they have this very classic shape, and it's illustrated in the picture, and that's the shape that we're used to seeing most viruses represented as. There is that top nuclear capsid head, which you can kind of see right here. That capsid head, which comes in a variety of shapes, actually will contain the nucleic acid inside, and we'll talk about that. That then goes down to this collar, followed by the sheath, and then the base plate and the spikes and the tail fibers. Remember, these tail fibers are what actually sort of detect the surface of the bacteria and tell the virus that it is a specific bacteria that they are interested in. These will then adhere to the surface of the bacteria and move in a way that pulls this spike and base plate assembly down, thus puncturing the cell wall and the cell membrane of the bacteria. And then the nucleic acids are injected from the capsid head down through the sheath and in through this base plate and everything into the bacterium. Now, there are two different types of sort of bacterial reproduction when we talk about them taking over their cells. There's either a lytic cycle or a lysogenic cycle, and these are the two sort of processes whereby viruses reproduce themselves with cells, so in bacteria. So let's talk about each of those. The lysogenic cycle is sort of like what we would call sort of the low-key, temperate, really non-virulent type of infection. What happens is that the phage will bind onto the bacterium here, and it will inject its DNA or RNA, depending on the virus, and we'll talk about that a little bit later, but it injects its nucleic acid into the bacterial cell. And that piece of DNA or RNA then goes and integrates itself into the bacterial genome. Here it's sort of shown as a circular plasmid, and becomes known as a prophage, okay? And that prophage can kind of sit there for generation after generation after generation and exist in the bacteria without actually creating a significant number of new bacteriophage particles. And this is sort of a waiting game. So with each cell division by the bacteria, and you can remember that on average bacteria replicate every 20 minutes to 2 hours, with each replication of the bacterium, the phage DNA is replicated, and so it's actually reproducing. But at this part, it's not making any more infectious viral or bacteriophage particles. So at some point, that prophage is going to say, we need to make more bacteriophage, right? It can't just all hang out in a dormant state forever. If the bacteria that the prophage is part of is actually subjected to some stress, whether that is nutrient deprivation or perhaps exposure to UV light or something, that prophage can detect that the bacteria cell is undergoing stress and sort of extricate itself from the bacterial genome. This is a process called induction, and it will begin to actively replicate. And as it actively replicates, it will then start to make more of the viral genome, and it will lead to synthesis of bacterial proteins, right? So that DNA is then transcribed into viral RNA, and that viral RNA is translated into the proteins that are then assembled to make more bacteriophage. And the way that this happens is that the prophage hijacks the bacterial cell. It uses all of the bacterial cells' polymerases, all of the translational machinery, the ribosomes, the endoplasmic reticulum, etc. It uses all of those things to manufacture only viral particles. And this eventually does two things. One, the bacterium can no longer survive because its natural processes are not taking place, but also the bacteriophage will fill up the entire bacterium and eventually break it open and kills that host cell. And then these new bacteriophage can float off to find another bacterium to infect. Okay, what about eukaryotic viruses? I know this is much more interesting to you because these are the kind of things that can infect all of us or our pets or the things around us. The first thing we should talk about is how eukaryotic viruses are classified. So there are several features that we look at when we're talking about these viruses. The first thing is, what type of nucleic acid do they carry? Do they carry DNA or RNA? And is that DNA or RNA single-stranded or double-stranded? And this will become a little bit more important later, especially the difference between DNA and RNA viruses, and we'll discuss that later in this lecture. Does the virus have an envelope, or is the capsid naked? And we're going to discuss that a little bit in coming slides. What is the shape of the capsid? Is it sort of a spiral or a tube? Is it more circular? Is it more of this sort of hexagonal, three-dimensional structure? Those sort of things are very, very specific to the virus. And then the last characteristic is, where in the host cell is the new virus made? Is the new virus made specifically in the cytoplasm, or is there some of the process in the nucleus? Okay, so let's talk about capsids. The capsid is the protein structure that contains and protects the nucleic acid that really is what the virus is. The virus is essentially an infectious piece of nucleic acid. The capsid itself is made up of single protein units called capsomeres. You'll remember from cell biology that we talked about how utilizing a single protein and assembling it in a specific way to make a very complex structure is a highly efficient use of genetic material. If you only have to encode one capsomere protein, and you can use that capsomere protein to assemble a highly complex structure, that is a real economy of the nucleic acid or the genomic material. And again, because these viruses are so small and you have to pack all of the nucleic acid necessary to replicate that virus into this capsid head, you want to minimize the amount of nucleic acid, whether that's DNA or RNA, that is present in that capsid head. So here we see two different types. We see the polyhedral type. This is that sort of hexagonal three-dimensional structure that we most commonly associate with bacteriophages. And here's an example of a helical structure. This one sort of resembles a tube in reality, with the nucleic acid assuming a helical shape within that sort of capsid tube. Okay. Now, not all viral nucleic acids are carried in a naked capsid. All viruses do have a capsid, but some of those capsids are actually enclosed further in a structure called an envelope. And interestingly, that envelope is derived from the plasma membrane of the cells that this virus infects. So let's go through this process. What happens is, if you see up here, the viral capsid has been assembled in this eukaryotic cell, and it has the nucleic acid inside of it, and it is going to go to the cell surface, and it's going to begin to push out of the cell surface and sort of bud off. And when it does this, that capsid is now protected by something that actually looks like that eukaryotic membrane. And it is that eukaryotic membrane. In some ways, we can think about this as a way that this not only protects this capsid, but it's a little bit of a cloaking mechanism. Now, it's not a complete cloaking mechanism because the virus then decorates the outside of this envelope with these spikes. And you've probably heard the term spike referred to several times when hearing about coronavirus, and we'll talk about that. These spikes are specific bacterial proteins that decorate the surface of the envelope. And in many cases, what they do is they serve as the recognition site. They will interact many times with a specific normal receptor on the cell to allow the virus to be recognized perhaps as a friendly protein or something good, and triggers the cell to actually take up the virus. One thing of quick note, enveloped viruses actually tend to be more fragile. If we think about the fact that this is a lipid membrane that is surrounding this capsid, and that this membrane is essentially made of lipids, just like our plasma membrane, these are easily disrupted by soap, right? We know that soap can actually disperse fats. This membrane or this envelope right now is essentially a lipid bilayer. And when that envelope is disrupted, this capsid becomes quite vulnerable. Okay, so we now have talked a little bit about the sort of physical structures. Let's talk about the types of nucleic acids that are involved in this. As I said earlier, a virus can either be a DNA virus or an RNA virus. Let's start with the DNA viruses. They typically have double-stranded DNA, but there are a few that do have a single-stranded DNA molecule contained in their capsid. And the process for DNA and RNA viruses to enter cells is fairly similar. I'm going to go through it here, but in this case, we're going to be referencing specifically DNA and what happens to that DNA when it enters the cell. So in eukaryotes, that virus is going to bind to a receptor on a specific type of cell. Remember, I mentioned the tissue tropism, and that tissue tropism is actually defined by the types of receptors on the cell surface. As we know, different types of cells sort of have a unique fingerprint of molecules that are on their cell surface. So the molecules and receptors and proteins on the cell surface of a cardiac cell are very, very different than those that are on the surface of a hepatocyte or a liver cell. So the virus will actually look for the receptor that it tends to bind to. And again, those spike proteins play a role in finding those specific receptors and binding to it. When those proteins interact and do actually bind to the cell, this will actually trigger the cell to take in that enveloped virus by receptor-mediated endocytosis. And so again, the cell it thinks that the receptor has bound to its specific protein of interest, and it begins to take that receptor ligand combination into the cell. Unfortunately, it's like a Trojan horse, and once the cell membrane has invaginated and surrounded and taken in that virus, it's almost too late. Alternatively, a subset of viruses can actually sort of bind to the surface of the cell and inject their DNA straight into the cell. If there is an envelope, when the virus enters the cell, that envelope is sort of removed, and then the capsid can inject the viral DNA into the nucleus of the cell. Certain naked capsids can also bind just to the surface of the cell and inject straight there. So there's a variety of mechanisms by which the virus can get the nucleic acid into the cell. The viral DNA then integrates into the host genome. It could sit there for a day, a week, several years, just waiting for something to happen. Or that DNA could interact with DNA polymerases in the nucleus. Those are the host's DNA polymerases, and immediately be replicated. And then from there, that replicated DNA can be picked up by the host cell's RNA polymerases, immediately transcribed into an mRNA, and then that mRNA goes out to the cytoplasm just like a eukaryotic mRNA would, where it then hijacks the host translation machinery and begins to crank out lots and lots and lots of viral proteins. And what typically happens is that this replication and transcription of the viral DNA and RNAs and translation of these so swamp out the cell's natural functions that the cell becomes sickly and unable to fight off the infection and will eventually die or is killed by the immune system. And one last note, some of these DNA viruses do actually carry their own polymerases with them, sort of associated with the nucleic acid as it's harbored in the capsid. So they don't necessarily have to go straight into the nucleus, to be replicated or transcribed. That DNA can actually sit in the cytoplasm and be replicated and transcribed by those viral-derived polymerases. And in that case, it absolutely takes over the cell. So let's talk a little bit about these DNA viruses. Here is a lovely schematic. This comes from your textbook, just a note, showing basically that we have a virus up here at the top. I'm looking for my cursor. And here this is, and it has a DNA virus here, and it has just a capsid here, or the coat protein. We're not showing this as an enveloped virus. That capsid binds onto the cell surface, and this is injecting the DNA directly in. We don't have any receptor-mediated endocytosis here, and this will then stay here in the cytoplasm, or this could potentially be in the nucleus depending on the type of virus. And it goes in two directions. This DNA can either be replicated to make more viral DNA, and again, either using polymerases that it brought with it, or by hijacking the host cell's polymerases, or it can actually come over here and some of that DNA can be transcribed into RNA, which is then translated into various proteins, especially these coat proteins, and this DNA and this RNA then sort of join up here down at the bottom to assemble these little viral particles. And as this occurs, the cell fills up with these viral particles until the point that it breaks open. One thing to note is that as this replication occurs of this viral DNA, this will occur so quickly that eventually this swamps out all of the RNA transcription machinery and the translation machinery. And so the viral DNA swamps out any of the processes with the genomic DNA and basically takes over the cell completely. So what are some examples of DNA viruses or diseases that you have probably heard of? Well, we have adenoviruses. These are fairly common. People get the variety of them throughout their lives. The pox viruses, I've noted them down here. Smallpox, which was eradicated shortly after I became an adult. That is no longer here because of vaccination. Chickenpox, most of you probably have been vaccinated against this. My generation, we all tended to get chickenpox, and chickenpox can reemerge when you are an adult as shingles. Again, we talked about how that viral DNA can integrate into the host genome, and certainly the varicella or chickenpox virus can do that. And when people are under particular periods of stress or there's something wrong, that virus can reemerge and we get a case of shingles. Herpes is a DNA virus, as is cytomegalovirus or CMV, which tends to cause certain birth defects, and hepatitis B. Okay. What are the most common types of viruses? Well, RNA viruses. Absolutely the most common. And really they come in two flavors. We have positive strand RNA viruses and negative strand RNA viruses. We do see single-stranded RNA viruses that are more common. There are a few that are double-stranded. The mode of entry into the host cells is very similar to the DNA viruses, but RNA does not necessarily have to be replicated or transcribed, and so it actually can hang right out in the cytoplasm and begin making more virus almost immediately by basically sort of hijacking the cell's ribosomes and translation machinery. Now, I talked about negative strand and positive strand RNA viruses. A negative strand RNA virus requires that the RNA must first be transcribed into an actual functional mRNA before it can be translated into protein, okay? So that specific RNA that is injected into the cell does not actually code for protein at that point. It has to be converted to an mRNA. And so that is important because that is a step at which we can look at developing antiviral drugs that could target that process. If we have a negative strand virus and we can prevent that conversion from the negative strand RNA to an mRNA, then that prevents the virus from ever replicating. So we actually have a target for therapy. So what is a positive strand RNA virus? Well, these are viral RNAs that actually function immediately as the mRNA. So as soon as the virus sort of offloads its nucleic acid, in this case, this positive strand RNA, into the cell, it can hijack the ribosomes and the tRNAs and all of the machinery necessary to make viral proteins. And this can happen very, very rapidly. And so as it goes through this, the replication of the RNA itself is sort of a secondary function that occurs after the positive strand RNA virus has staged an entire takeover of the cell. And the fact that these do not have to go through that step of converting to mRNA actually makes these positive strand RNA viruses particularly problematic. The infection is fast, and there is not a step that could be targeted maybe to interrupt the process as a therapy. One thing you guys should know is that SARS-CoV-2, which is the virus that causes COVID-19 right now, it's a positive strand RNA virus, and that is not good. So what are some diseases caused by RNA viruses? You've all heard of Ebola. I know you guys like to talk about it during class and what happens to people and all of that sort of stuff. Polio, which you've all been vaccinated against, is also an RNA virus. SARS, which is severe acquired respiratory syndrome. This was the original respiratory coronavirus that emerged in the early 2000s. It was restricted primarily to the Asian continent, but made people profoundly ill. And eventually that pandemic ran its course, and that virus has not reemerged. You will sometimes see that one referred to as SARS-CoV. COVID-19. So COVID-19 is the pandemic that we are dealing with right now. COVID, which stands for Coronavirus Identified 2019, is caused by the SARS-CoV-2 virus. It is very, very strongly resembles that original SARS-CoV virus from the 2000s, but it causes a much more significant respiratory issue. MERS is Middle Eastern Respiratory Syndrome. This is a relative of SARS and COVID-19. that emerged, I think, in the Egyptian area back around, was it 2010 or so, and is a very difficult disease to deal with, and that has mostly sort of gone to the wayside. Hepatitis C, yearly influenza, we all get our flu shots, and measles and mumps sort of round out the RNA viruses. Now, with RNA viruses, there's actually a special class of these that we call retroviruses. They're very much a special type of RNA virus, and it is because the nucleic acid, or the virus, carries with it an enzyme called reverse transcriptase. Now, this enzyme is absolutely exploited by molecular biologists like myself. Reverse transcriptase will take a piece of RNA and convert it to a DNA molecule, right? So we've talked about the central dogma of molecular biology, which is the fact that DNA is transcribed to RNA, which is transcribed to protein, and that in general, these steps are unidirectional. They don't really go backwards. So protein is never converted back to RNA, and RNA in general is not converted back to DNA, except with an enzyme like reverse transcriptase. And so in the laboratory, we can actually use reverse transcriptase to make a DNA copy of an RNA of interest. And again, this comes from these retroviruses, and that's where we isolated it from. So in a retrovirus, the virus injects its RNA into the host cell, and it can either be a positive or a negative. And then coming along with that nucleic acid into the cell is the enzyme reverse transcriptase. And that then converts the viral RNA to DNA, and it goes backwards of the central dogma, right? The DNA then goes to the nuclease, and it'll integrate into the host genome. And when the cell divides, that viral DNA is replicated with the host DNA and is thus carried to the daughter cells. In the next slide, we'll kind of go over a little bit of a specific example of a retrovirus and what this looks like. But some examples are things like HIV, human immunodeficiency virus, which we know is the virus that underlies AIDS. There are feline leukemia viruses. For those of you who have cats, we vaccinated our cats against this. And we also worry that our cats might be FLV positive because they can infect other cats. That leukemia is caused by a virus. And there are actually leukemias in humans that are caused by viruses. Human T-cell leukemia viruses, also known as HTLV1 and 2, both lead to the emergence of leukemias in humans because when they integrate into the genome, it is a very targeted integration into the human genome, and it interrupts genes that are associated with suppressing cancer, and this leads to the emergence of leukemia. So let's talk specifically about these steps with retroviruses, and we're going to use HIV as an example. This is a beautiful diagram showing each of these steps, and we're going to sort of go through them briefly. But the first step is that HIV fuses to the host cell surface. Now, something to recognize here is we have HIV. This is the virus. You'll notice that it is an enveloped virus. The RNA is sitting happily in here inside this capsid. And we have these little spikes here on the outside that are decorating that envelope and helping the virus locate the specific cells it's interested in. The receptor that this tends to look for is something called CD4. And this receptor has a co-receptor called CCR5. Why am I pointing this out? Well, you'll remember in our bioethics discussions that we talked about the scientist in China who had done the CRISPR knockout or rendering non-functional the CCR5 gene in the twins that nobody has been able to locate. And so his target actually was this CCR co-receptor, which he made a CRISPR modification to, hoping that if he rendered this co-receptor non-functional, that these cells would no longer be susceptible to HIV infection, okay? We have discussed ad nauseum sort of the ethics of all of this, and so I'm not going to go into that here, but I wanted you to be aware that we are talking about this system that we discussed related to CRISPR. So the HIV fuses to the host cell surface, it comes over here, and it now sort of triggers this binding or sort of the fusing of the envelope with the surface of the cell, and into the cell it releases the capsid, and the capsid contains with it the RNA. It contains the reverse transcriptase because this is a retrovirus. It also carries a protein or an enzyme called integrase and a few other viral proteins. And this all gets dumped into the cytoplasm of the cell. So once this RNA is dumped in here, that reverse transcriptase immediately converts that RNA to a DNA, okay? And so it's shown here that you can see the viral RNA is being reverse transcribed into the DNA molecule, which is this slightly darker red color. It then is joined by this integrase enzyme, and that integrase enzyme actually helps sort of put the viral genome into the host or the human's genome, and it integrates in there. Very, very similar to our bacteriophages, where when integrated into the host bacterial genome, it is called a prophage. In this case, when we have the viral DNA residing in the human genome, it's integrated in, we call that a provirus. Now, this provirus can actually sit sort of dormant for a little bit, but in this case here, it's showing that this now is transcribed to make new viral RNA. And this piece of DNA is selectively transcribed. It has very, very active promoters and recruiting elements that pull a large amount of the host cell's replication and transcription machinery to this spot. And so the cell, the situation favors making new viral RNA. That new viral RNA then goes out of the nucleus through these nuclear pores. I'm not sure why they're showing it going against the nuclear membrane. But once that RNA is out into the cytoplasm of the cell, it is translated. It is used as the template in which to make a variety of the viral proteins that are necessary. These viral proteins will actually join up with some of this replicated viral RNA, and these will assemble into a new viral or virion structure. And so here we're seeing this mature further. It is beginning to develop and form its capsid. It is forming this envelope as it is pushing out of the host cell membrane. And once it buds off, we now have an enveloped or enveloped capsid-bound RNA molecule containing some RNA, some reverse transcriptase, some integrase, and the virus has again decorated its surface with these spike proteins. Okay, so that's going to conclude this lecture on viruses. I know this is a lot of information to take in, but this is really important information, not only for understanding aspects related to your own healthcare and what is happening around us in terms of the development of these viruses that can lead to pandemics, but also in terms of understanding these as a molecular biology tool. Viruses are very commonly used in the lab. They were one of the first sort of mechanisms that were tried out for gene therapies. They were not particularly successful, but they do have uses. This will be followed up in a day or two with another lecture that will deal specifically with coronavirus and how we are studying that. So if you have any questions, you know how to find me on our Slack class page, please post them there because your classmates may have the same questions. And I will look forward to seeing you in the next round. Oh, and by the way, on Slack, I have posted kind of a fun bacteria/virus activity for you guys. It is a virtual escape room, and you're going to need to use your new knowledge of viruses and some of that old knowledge of bacteria and their genomes in order to get out. Guys, take care, and we'll see you soon.